Hey everyone, welcome back to Windows Seed Studies. I hope you all are doing well. So, we were discussing about neurobiology, in which we have covered neurons, the structure of neurons, types of neurons, as well as electrical properties of neurons, which makes the neurons the signaling unit of our nervous system. We also discussed about neuroglial cells, how the different types of neuroglial cell myelinate the axons that are present in our peripheral nervous system as well as in the central nervous system. So today's lecture is the continuation of the previous lecture where we were discussing about neuromuscular junctions. So neuromuscular junctions, they are well studied example of chemical synapse because they are very easy to study due to ability of inserting multiple microelectrodes in the muscle fiber. Okay, so we in the last lecture we learned about how muscle fiber and nerve terminal they form a synapse and how the acetylcholine neurotransmitter is released from the presynaptic membrane and that is received by the postsynaptic membrane through the acetylcholine receptors. And in this lecture we are going to learn about how different researchers they experimented and gave out the different results. Like the when we were learning about action potential generation, we learned that the property of neuronal membrane being polarized makes them excitable, and when signal arrives, when stimulus is received on this membrane, some ion channels open and that changes the voltage of the membrane, which further opens the voltage gated ion channels, changing the membrane potential. And this is how action potential is generated. Okay, so this action potential travels over the axon, and this is how nerve impulse is conducted. So when we were learning about action potential, we already learned about which type of ion fluxes are being generated, like sodium, the movement, the direction of movement of sodium ions as well as potassium ions. But in the today's lecture, we are going to learn about the experiments that made the researchers to make those conclusions. So we already know that the cell membrane, the neuronal membrane is polarized. It has a higher concentration of potassium ions inside and higher concentration of sodium and chloride ions outside. Well, there are anions on the cytosolic part, which gives the negative charge towards the cytosolic part of the membrane and the positive charge to the extracellular part of the membrane. Okay, then we learned that the in action potential, the whenever signal arrives, the local depolarization happens. Means this polarized state, this polarization is reduced. So reduction in polarization means reduction in charge separation is due to the movement of the sodium ions. Okay, but movement of sodium ions is seen, but this doesn't exactly reach the change. The charge separation doesn't exactly reach to the equilibrium potential of sodium ions because there is some efflux of potassium ions as well. Okay, so we learnt about this in the lecture where we discussed about action potential generation. So if you don't know about the details, please refer to that lecture. So we already know that the membrane potential doesn't reach exactly to the equilibrium potential of sodium ions, but it reaches somewhat closer to that, and that is what we call as action potential. Once action potential is generated, it is propagated over the axons. But what kind of experiments scientists did to give this inferences? We haven't learned about that yet. So in this lecture, we are going to see what kind of experiments were done on the neuromuscular junctions, because all of those studies were done on the neuromuscular junction, and then scientists gave the detailed process of action potential generation and propagation. So we are going to learn about those experiments. We have already discussed about patch clamp technique. If you don't know about it, I'll revise it in short. Okay. So in last class we found out, like we learned about the experiments that were done, the intracellular voltage recording experiments that were done by Paul Fat and Bernard Katz. So they used the Curer drug, the plant toxin, to isolate the end plate potential. So how did they isolate the end plate potential? They reduced the postsynaptic potential below the threshold value for the action potential. And that is how they found out that this excitatory postsynaptic potential, which we refer as the end plate potential, this decreases progressively over the distances. Okay. So that was what we learned in the last class. So in that class we discussed about patch clamp technique as well. So in the lecture where we discussed about equivalent circuit model, we already discussed about Ohm's law. So in that lecture, we learnt about the analogy between the electrical circuits as well as the neuronal membrane. Means we've considered this lipid bilayer, this neuronal membrane, as an electrical circuit, like how the ion fluxes are being generated, and they are they are generating ion currents, and small electrical circuits are being formed. So in that lecture, we learned about the analogy of lipid bilayer with a perfect resistor whose resistance is decreased due to presence of ion channels, and the membrane also has capacitance, and it also has electromotive force due to the unequal distribution of ions. So if you don't know about that, please refer to that lecture because it is a kind of prerequisite for this lecture. So in that lecture, we learned about Ohm's law. You have already learned about Ohm's law in your physics classes in high school, but let me just revise it. So Ohm's law says that whatever current is generated, so here we are mentioning about the postsynaptic current, like postsynaptic current, the current that is generated on postsynaptic membrane. So I'm going to write it as excitable postsynaptic current. Okay, so EPSP means whatever is happening, the changes that are being made on the postsynaptic membrane. So current that is generated on any membrane, here in this case there is postsynaptic membrane, that is always equal to the conductance of that membrane into the electromotive force that is being that is deriving the ion fluxes. So that will in this case it will be equal to the conductance of the postsynaptic membrane into the membrane potential. Okay, the membrane potential of the postsynaptic membrane minus the equilibrium potential that is being generated due to the unequal distribution of ions across the membrane. Okay, so this equilibrium potential, this is due to the sodium ions. Okay, this is due to potassium ions, this is due to chloride ions. So we are taking into consideration all of those ions. So we learned about Goldman equation. So Goldman equation gives us the idea about what should the membrane potential be considering the equilibrium potential of those particular ions. And Nernst equation gives the value of those particular equilibrium potential. For single ion, you use the Nernst equation. For considering all of the ions, like how those all of the ions and their permeability to the membrane, how do they contribute to the membrane potential, that is what is derived by the Goldman equation. Okay, but Goldman equation didn't give us the idea about the dynamic changes that happens in the neuronal membrane, and that is why we learned about the equivalent circuit model. Okay, so please refer to that lecture. I've discussed all those things in detail. So the Goldman equation, so you can have the values of those the individual E's by the Nernst equation, and the value of this by the Goldman equation. Okay, so this is the chemical driving force, like the driving force which is being generated due to the electrochemical gradient of the ions. Okay, that are unequally distributed across the membrane. So this equation is very important. This Ohm's law, I have discussed it in this previous lecture as well. This is very important because using this, the researchers were able to deduce that how the action potential is generated, and they were able to deduce that the acetylcholine receptor that are going to generate both sodium ion currents as well as potassium ion currents. Okay, so in the depolarization, so we already knew before doing those studies, we already knew that the chemical concentration gradient for the sodium ions were in this direction. Means the concentration of sodium ions was high on the extracellular side and low on the intracellular side. So this gives us the idea that the ion current, the sodium ion current, should be in this direction. And when it is in this direction, the polarization is decreased and membrane gets depolarized. Okay, so by theoretically the equilibrium potential that should reach to the membrane potential should reach to the equilibrium potential of sodium ions, but this does not happen. This means that there is some potassium flux as well. Okay, so today we are going to see how this was concluded experimentally. Okay, so theoretically, let's see what should be there. So whenever the membrane potential, this gets, so theoretically this should get equal to the equilibrium potential of sodium ions, which in this case is the plus 55 millivolt. Okay, the equilibrium potential for sodium ions is plus 55 millivolt. This value is derived by Nernst equation. Okay, this is Nernst equation uses the ion concentration. So Nernst equation tells us about, like, the equation is given by RT by NF into log of the concentration of potassium ions outside the cell divided by concentration of potassium ions inside the cell. So here in this case we are deriving this value for sodium ions. So let me write the sodium ion concentrations. Okay, this gives us the value for the equilibrium potential for the sodium ion. So theoretically, that this membrane potential should reach to the equilibrium potential of sodium ions when the depolarization happens, because all of the sodium ions will move to the inside, okay, and will make the whatever unequal distribution was there, they will make that equal. So let's say that there are 3 sodium ions here. I'm just taking an example, and 2 sodium ions here. So what is the concentration gradient? Concentration gradient is from outside to inside. So one sodium ion, let's say that one sodium ion moves from extracellular matrix to inside. So this will reduce the number of sodium ions here. It will make 2 sodium ions here, and let's say 3 sodium ions here. So this is making the membrane which is inside positively charged, you know, compared to the outside extracellular part. Okay, so this is what should happen, but this does not happen exactly, because there is some, so this tells us that the membrane potential should reach to the equilibrium potential of sodium ions, but this does not happen. The membrane potential does not get exactly equal to 55 millivolt, because there is some other current is being generated as well. So how was that deduced? Because let's say when the Vm gets equal to the equilibrium potential of sodium ions. So let's say that the only sodium ions, hypothetically, we are forgetting about the potassium ion currents. We are only taking a look at the sodium ions. So let's say only sodium ion is moving from outside to inside. So whatever equilibrium potential is generated, it will be due to sodium ions, because the current, the ion fluxes, which ion is getting, there is only one ion which is moving, and that is sodium ion. So the the postsynaptic potential, equilibrium potential of postsynaptic membrane will be equal to ENA. Okay, so when it is equal to ENA, let's say that we are changing the values of the, so when once the membrane potential gets equal to VNA, okay, at that point of time the ion current should not be generated. So I should be, because this value mathematically this gets to 0. If this is 0, then the current to generation will be 0. So the potential at which net ionic current is 0, it is referred as the reversal potential for that current. Okay, let me explain it in a different way. So ion, so let's say that the current generated is only due to the sodium ions. Okay, only due to the sodium ions. So that will be equal to the conductance of the postsynaptic membrane to the sodium ions. Okay, into the membrane potential minus the equilibrium potential of sodium ions. So this is in the hypothetical case when we are considering that only sodium ions are moving. So only sodium influx is seen, and that is depolarizing, and that is depolarizing the membrane. But we already, I was discussing about action potential generation, I told you that in the depolarization of membrane, not on there, alongside the sodium influx, there is potassium efflux as well. But now I'm going to tell you how that was derived, that was proved experimentally. So if this is the hypothetical case, okay. when we are saying that only sodium ions are moving. So only, if only sodium ions are moving, then the current generation will be only the sodium ion current. Okay, sodium ion current will be equal to the conductance of the membrane for sodium ions into the membrane potential minus the equilibrium potential of sodium ions. So we proved, we wanted to test this experimentally. So this membrane potential can be changed. How can we change the membrane potential? Let's say that this is a muscle fiber and I'm introducing a micro electrode here. Okay. So if if you introduce if you this is a voltmeter. Let's say to this a voltmeter is attached. Okay, voltmeter is attached, which is going to measure the membrane potential as well. So when you insert the micro electrode, you and you change the membrane potential. So you are changing the membrane potential. So this membrane potential is, let's say that you set the value exactly equal to the equilibrium potential of sodium ions. So we know that this value is plus 55 millivolt. Okay, by we calculated this theoretically. So then let's say that I set the voltage of the membrane potential, like I set the membrane potential through using the voltage apparatus. I set this to the plus 55 millivolt. Okay, so once I equalize both membrane potential to equalize the membrane potential to the equilibrium potential of sodium ions, this will get to zero, and this means that no ion current will be generated. So there will be no ion current, no sodium current. Okay, sodium current is equal to zero. This means that sodium ions is not moving. So the potential at which net ionic current is zero means this potential, this plus 55 volt, the value of potential at which the ion current for that particular ion is zero. This is known as reversal potential. So this is the reversal potential. Okay, reversal potential. This value plus 55 volt is the reversal potential for sodium ions, reversal potential of postsynaptic membrane for sodium ions. Okay, this is the reversal potential of postsynaptic membrane for sodium ions. But this value will be different for potassium ions. Okay, and this value will be different if the ion current is generated by sodium ions plus potassium ions. Okay, so let's say that we set the so let's take a look at what were the results when the scientists they set the membrane potential to the 55 millivolt. So I have a diagram for those cases. So here is the diagram. I've taken those from the Principle of Neural Science book, which written by Eric Kandel. Okay, so in the hypothetical scenario, when I set the voltage value, the membrane potential to the 55 millivolt, this means that there will be no current, no sodium flux. So there will be no movement of sodium ions since the current is the zero. INA is zero at the 55 millivolt potential. Okay, so once I decrease the value of membrane potential, means at the rest, when the cell is at rest, it has minus 90 millivolt value. The current is negative. Negative current means, okay, so current is a scalar quantity, it cannot be negative, but here in this direction, conventionally in electrophysiological experiments, negative current means the direction of current is inward from outside cell to the inside of the cell. This means whenever current is negative, that it is only representing the direction. So let's say that the current is minus 3 picoampere. Okay. When I say this in the biology that current is minus 3 picoampere, so this doesn't make this doesn't make sense in physics because current is a scalar quantity. You cannot assign a negative value to that. But whenever I say this in electrophysiological experiment, it means that the amplitude of current is 3 picoampere and negative sign here shows the direction of the current, and conventionally this direction negative means the direction is from outside towards the inside of the cell. Positive 3 picoampere current means that the direction of current is from inside to the outside of the cell. Okay, so this is by convention. So when the cell is at rest, in the last class you already see how the electrophysiological, like the voltage recording, are the voltage recording that done by Paul Fat and Bernard Katz. You already saw that in last lecture. So at when the cell is at rest, means at the minus 90 millivolt, the negative current, some negative current is generated, right? This means that the sodium flux is from outside the cell to the inside cell, and this is because of the concentration gradient. This is obvious because on the outside the concentration of sodium ions is high, on the inside it is low, so sodium ions should move from outside to inside, so current generation is in negative value. Okay, so but when it reaches to 55 millivolt, no movement of sodium ion will be seen, because now the concentration gradients, the concentration of sodium ion which is outside and concentration which is inside, this is exactly equal. And when the two sides have equal concentration of sodium ions, no sodium ion is going to move across the membrane. Okay, so this is, and then when you increase the membrane potential to a more positive value, the positive current, means sodium outflux, efflux will be seen. So this is hypothetically, this is what result should be when you do the voltage recordings, voltage clamp. You use the patch clamp technique for do the voltage recording. Okay, but this wasn't seen. What was seen was this. This was the actual result of the study. So they saw that at when the cell was at minus 90 millivolt, it showed some negative current, means there was inward current generation. Okay, then they saw that at the 55 millivolt, where the current should be zero, it showed a positive current. This means there is efflux of some ion. Okay, but theoretically and logically, sodium ion should not move, because concentration gradients is zero for sodium ion, means the concentration of sodium ion outside the cell and inside the cell, that is exactly equal. But there is some positive current. This means instead of sodium ion, something is moving outside the cell. And this is how scientists made the conclusion that potassium efflux is seen at 55 millivolt. Okay, this means that the whatever ionic current is being generated, that is not only due to sodium ions, but it is due to both sodium ions as well as potassium ions. So they saw that at 0 millivolt, okay, when the membrane potential was 0 millivolt, at that point of time the equilibrium potential was zero. This means that the efflux, this happens when the potassium efflux is exactly equal to the sodium influx, means there is no net movement of ions. Whatever is moving out, exact amount of charge is moving in, because both potassium and sodium, they have plus one charge, right? So whatever charge is moving out, that is exactly moving in. So even though there is ionic current is there, there, even though there is ion flux, but ion current is zero. Okay, because the ion flux of the opposite side gets equal, so whatever efflux is there, that is exactly equal to influx. This means ion current, the net ion current I is equal to zero, because INA is exactly, it is exactly opposite to I potassium. Okay, I hope this is clear. So this is how the recordings, the voltage, intracellular voltage recordings were done, which gave us an idea that the generation of action potential is due to both sodium ions as well as potassium ions. Okay, so the reversal potential for both, so when reversal potential for sodium ion is plus 55 millivolt, but reversal potential for sodium plus potassium ion is 0 millivolt. Okay, I hope this experiment is quite clear to you all. Okay, then let's learn about patch clamp recordings. Okay, what were now we are going to see the experiments that were done by Erwin Neher and Bert Sakmann. So we are going to learn about experiments done by Erwin Neher and Bert Sakmann. Okay, they did those experiments in 1976, and they made some inferences. So let's first see what kind of experiments they did and what were the inferences made by them. So they used a patch clamp technique to measure the flow of ionic current that happens through single acetylcholine receptor. And how did they manage to do that? So there is something called patch pipette, which is basically a glass micropipette, which that is inserted on the postsynaptic membrane. In this case, we are studying about muscle fiber. So our muscle plasma membrane is the postsynaptic membrane in this case. So the patch pipette, the glass pipette is inserted and it pinches off a part of the membrane. Okay, you can see it is tightly sealed here. So it pinches a part of the membrane. So it is electrically separating. We are electrically separating a membrane, and that is why these are known as patch clamp techniques. Okay, and using this patch clamp technique, the current that is generated through the acetylcholine receptor was recorded, and some inferences were made. So they separated some part of the membrane. Okay, and when they did this patch, this glass micropipette has some solution. Uh, sorry. So it has some solution in which there is some solution. So in this case, there was salt solution. So this is referred to as the reference. This is referred as reference or ground state. Okay, so usually in the voltage experiments, when you have to change the membrane potential, a microelectrode is inserted, which is going to provide some current as a stimulus, and that is going to change the postsynaptic membrane potential. But in this case, you have to learn about the signaling that happens at the nerve and the muscle synapse, right? And how does that happen? That happens through acetylcholine, and that is why in this case, the both of those scientists, they use acetylcholine in that salt solution, because acetylcholine is going to bind to the acetylcholine receptor, and that is going to induce the signaling. Okay, so in the case, let's say that instead of, you are not learning about the postsynaptic membrane is not muscle cell. Let's assume that it is nerve cell. Okay, and you have to measure how the electrical activity goes in the nerve cell. So at that point of time, you know that it happens due to voltage gated ion channels. So you are going, instead of providing acetylcholine, you are going to change the voltage of the membrane by introducing microelectrode, and that is how you do the experiment, and that was done by the fat and cats that we learned in last class. Okay, but in this case, you are learning about chemical synapses, right? That is why you are using acetylcholine, because it is a ligand which is going to open a ligand gated channel, which we call as acetylcholine receptors. And now the ion channel opens, which does some ion influxes, and that generates the action potential. Okay, so they both of them, they concluded after doing certain experiments, they concluded that opening of an individual ion channel, this generates a small rectangular step of ionic current. So when they introduce acetylcholine, and once acetylcholine binds, so they measured through this apparatus, they measured the current which is generated. So when they measured that current, they concluded that, like, they observed that whatever is being generated, it was very small. So the current that is being generated is very small, and it is a rectangular it is a rectangular step of ionic current. Okay, well, this current is very small, but this is being generated due to the opening of ion pores that is letting through millions of ions. Okay, it is doing ion flux of millions of ions. We'll learn about the statistics as well. Okay, so they saw that the small rectangular step of ionic current is generated. Let me show you the recordings. So these are the recordings that were done by them, and these are published in the same book, The Principles of Neural Science. Okay, so they saw whenever channel is open, so a small rectangular step of current is generated. Here you can see again the current is negative, means the inward current is being generated. Here you can see this is minus 2 picoampere. Okay, so like from the baseline to above, this is 2 picoampere, means that whatever current is being generated, it is average to minus 2 picoampere in these cases. So you can see small and rectangular ionic currents are generated. But when they what does this diagram tells you? This tells you that opening of this ion channel. Okay, the opening of the ion channel is like it is random, like whenever the ion current is sorry, the stimulus, the what we call ACH, acetylcholine, whenever it is binding to that, the opening of ion channel is at different points. It is at at different times, and the duration for which the ion channel is open, it is again different. Okay, so the current that is generated through single acetylcholine channel is same. So the current, value of current, here you can see in each case the value of current that is being generated, it is almost same. It is average to 2 picoampere, minus 2 picoampere. Minus means the current is inward. So the current generated through single acetylcholine channel is same for every opening. See, here are multiple openings. For every opening, it is same. But the duration for which the ion channel is going to open and the time between opening of two channels, that is different. So here you can see one ion channel is opened, and after t2 seconds, this next ion channel opened. Okay, here you can see the first ion channel opened, after t1 second, the second ion channel opened, and we know that t1 is not equal to t2. Right. Then again, for the duration for which this ion channel was open and the duration for which those ion channels are open, that is different. So the whatever duration and whatever time of opening is, so time of opening and duration of opening of ion channel doesn't matter. Whatever it may be, whatever value of time and duration for opening of ion channel may be, but whatever current is being generated, that is always equal. And this was a significant discovery made by them. Okay, so this was the very first reference inference, sorry, that was made by them, that the channel, sorry, the current generated through, well, this is done by, like those recordings are done for single ion channel, single acetylcholine ion channels, because patch clamp almost is separates the single acetylcholine receptor. Okay, so the current generated through single channel means through the single acetylcholine receptor. Okay, it is same for every channel, for each. So like they did this for multiple channel na. So they observed that current generated through single channel is same for every opening. It doesn't matter. Okay, but the time and duration of each opening varies considerably. So you can see that the so this is again, this is not possible. Like it is not really possible that each ion channel opens at, because so let's say that this there are six ion channels. Okay, okay, and to which acetylcholine is binding. Okay, so you cannot really refer that opening of all those six ion channels is at the same time, because the acetylcholine may bind earlier to the channel 1 and after some while to the channel 3. So the ligand binding is, it is happening at different times, and that is why the opening of ion channel is again happening for like different time frames. And again, the duration for which an ion channel, single acetylcholine channel opens, that is also not same, because first there are two binding sites on acetylcholine receptors for acetylcholine. So both of the binding sites should be occupied by acetylcholine. So some channels to some acetylcholine receptor, acetylcholine binds for longer duration. Acetylcholine esterase degrades that acetylcholine for after some while, and that makes the ion channel open for a longer over time. So duration again is not fixed for it is not same for all ion channels, but even the time and duration varies. So like logically you should say that if the ion channel that is open for 2 seconds, and let's say ion channel that is open for 5 seconds, so like you should, yeah, you might conclude that ion channel which is open for 5 seconds, it should generate higher current because more number of ions are going to move through. But this is not the case. Even the time and duration of each opening varies, the current whatever is generated, so current which is generated by the ion channel which opens for 2 seconds and the ion channel which opens for 5 seconds, both of them they are going to generate same current. And this was a very interesting discovery made by those researchers. Okay, so this means that a single acetylcholine receptor is going to generate same amount of current no matter the time and duration of the opening. So this is usually it opens for 1 millisecond usually, and it leads through the ions, almost 17,000 ions, 17,000 sodium or potassium ions. Okay, we already saw that it leads through both sodium ions as well as potassium ions. Here in this experiment, we already saw that the both potassium ion flux and sodium ion flux is seen. This again proves the point that the acetylcholine receptor, it is not, it is not selective for single ions. Unlike the voltage gated ion channels, the voltage gated sodium ion channels only leads through the sodium ions. Voltage gated potassium ion channels only leads through the potassium ions. So like voltage gated sodium ion channels will not allow the efflux or influx of potassium ions, and same goes for the voltage gated potassium ion channels. But this is not case for acetylcholine receptor. Acetylcholine receptor, it leads through potassium ions as well as sodium ions. This was seen, this was already proved by this experiment, because the postsynaptic membrane equilibrium potential is basically the due to the acetylcholine receptor, right? Only one single type of ion channels are present on the muscle fiber, and that those are the acetylcholine receptor. And that is why again this makes the study of NMJ easy. Well, so when the advancement were done in the electrophysiological techniques, certain electrophysiological measurements told about the pore size of the acetylcholine receptor, and the pore size of acetylcholine receptor is 0.8 nanometer. Okay, and this is quite large for letting through the sodium ions as well as potassium ions. So that is why those receptors, they are not selective for single ions, but yeah, they do not let through the anions due to the negative charge that are present on the, that the negative charge that are present on the ions, the anions. So ions with negative charge, they will not pass because they have negative charge. And why does they not pass? We'll learn about that in next lecture. Okay, so but they only lead through positive ions, no matter it is sodium ion, potassium ion. And then it was seen that 0.8 nanometer large pore is enough. It also leads through the divalent ion, the calcium ion. Okay, so as through the acetylcholine receptor, all kind of cations that are present on the in the neurons and the extracellular matrix, they can pass through, but it won't lead through an ion. Okay, this was experimentally proved by this, and then electrophysiological measurement also support this theory that the acetylcholine receptors, they are cationic receptor, cationic ion channels. Okay, so then the where were we? Yeah, so this diagram tells us about that the current generated through single acetylcholine receptor is same. Then this is the histogram that is, that is generated after the after taking the measurements of the after through the opening, like what is the value of the current which is generated after opening of the receptor. So that summed up to the, like, it had a normal distribution. This is a normal distribution curve. Okay, so that whatever statistical data is given to you, it is normally distributed, means it has only single peak, and that peak came out to be minus 2.7 nano ampere. Well, even we say that the current which is being generated, it is same, but it can vary with small, like with small numbers, with small value, due to the noise, due to some biological change, like biological phenomena. Okay, because there is always some error. So even though it is fluctuating in certain range, but it has a peak. This means it is normally distributed. So from this they concluded that acetylcholine receptor, the current that is being generated through single acetylcholine receptor, it is minus 2.7 picoampere. Okay, again minus means the current is inward. Okay, so why is current inward? Let me show you that in this diagram. Here you can see when you know whenever during like when the cell is at rest, the total current is inward at resting membrane potential minus 90 millivolt. The current is inward, but this doesn't mean that there is no ion current outward cell. So whatever the like total value is this minus this. Okay, the influx minus efflux. Okay, so like initially we thought that there is only inward current, but on with after some experiments we found out that there is influx as well as efflux, but this totally like the net current is inward. Okay, so whenever I say that current is inward, this doesn't mean that the only influx is happening in the cell. This means that the net current is inward, but yeah, there is some efflux. We already saw that there is potassium ion efflux and sodium ion influx, but net current is inward. This means that sodium ions are the ones that are that are giving the inward current, and that is why when remember when we were learning about action potential, we said that whatever action potential is being generated, that is due to the influx of sodium ions. Okay, but this does not exactly reach to ENA due to some efflux of potassium ions. Okay, so I hope you are now able to relate all of the information that we learned in that class. So this tells us about the current, the value of current is minus 2.7 pico ampere. And this normal distribution of this curve, this also tells us that only single type of acetylcholine receptors are present. Okay, so this means that whatever one acetylcholine receptor is doing, all of the acetylcholine receptor has same activity. Okay, so this is this is represented by the normal distribution of this curve. Okay, yeah, this is again the other observation made by Neher and Sakmann. They saw that whenever you generate current, when you give multiple stimulus, the current generated can be summed together. Okay, here you can see the summation of current. This tells us that, so this is not due to, this is not due to the single ion channel. So the first receptor, let's say opened, okay, and that is generating 2 ampere current. But alongside this single acetylcholine receptor, the second receptor is also opening and third receptor also opens. So the ion current that is generated by multiple ion channels, that can be summed together. Okay, so summation of ion current which is being generated by multiple receptors, that can be summed together. This was, this was proved by those scientists. This means that whatever ion current is being generated, okay, by single ion channel, let's say that ion current that is being generated, okay, well, this is due to both sodium influx and potassium efflux. So this is I, the small I means the current that is being generated through single acetylcholine receptor. Okay, do not forget that. And the capital I, when I write this is the total current. Okay, so like it's not like in the muscle fiber only single acetylcholine receptor is open. Multiple acetylcholine receptors are being opened due to the ligand binding. So that when the multiple acetylcholine receptor opens, whatever current is being generated, that is the capital I, the total current. Okay, total current of postsynaptic membrane, and this is the current which is generated by the single acetylcholine receptor present on the postsynaptic membrane. So this is equal to the conductance. Okay, conductance. Okay, let me represent it this with this value, with this symbol. So this is the conductance of the single, conductance. of the membrane, of the postsynaptic membrane due to single acetylcholine receptor. So you are imagining a scenario that on the membrane only one acetylcholine receptor is present. Okay, so like this, so membrane, if let's say that this is the membrane lipid bilayer which has no ion channel, no acetylcholine receptor, this will have resistance, infinite resistance. But due to presence of this ion channel, the resistance decreases to some finite value. Or we can also say that the capacitance of, sorry, the conductance of this lipid bilayer which has no ion channel is zero. Okay, because the conductance is inverse of the resistance. So this means that the conductance of this membrane, the membrane 2, the membrane B, this, let's say this is membrane A and this is membrane B. The membrane B, which has one acetylcholine receptor, the conductance of this membrane will have some value and which will be higher than the conductance of A, because, well, A is 0, right? So the kind of value of the conductance of the membrane B will be higher than whatever membrane A has. So, but if I increase acetylcholine receptor, conductance is going to increase. Okay, and that will make a conductance G. So this G is conductance of the membrane. Okay, let's say that here we were discussing only a membrane with single acetylcholine receptor, right? So the membrane B is the one with single acetylcholine receptor. But in this scenario, we are discussing about membrane with multiple acetylcholine receptors. This is membrane C. Okay. So the conductance of this membrane is gamma. This is due to single acetylcholine receptor. Okay. Multiplied by whatever electromotive force is being generated, that will be the membrane potential minus the the equilibrium potential of postsynaptic membrane. Okay, this value is same, because this value is due to, this value, the equilibrium potential will be same for this as well as this, because this is derived due to the unequal distribution of ions. So the distribution of ions is not going to change. We are just changing the membrane property. Okay, so but in this case the value will be G of the postsynaptic membrane into Vm minus E of postsynaptic membrane. Okay, so after the experimentation, they saw that when this, this experiment, told them that when the single ion channel is opening, whatever current is one acetylcholine receptor generates, like let's say that this is generating I current, this is generating I current, this is generating I current, and here we see that I1, okay, I1 will be equal to I2, will be equal to I3, because each acetylcholine receptor, okay, this is I1, this is going to do I2, this is going to do I3. So we already saw that each acetylcholine receptor is generating same current. So I1 is equal to I2 is equal to I3. We already made this conclusion from the previous recordings. Okay. But what researchers saw after this was that all those ion currents, they can be summed up, I1, I2 and I3, which are equal. So let's say that I1, I2, I3, all of them are I. So, okay, so the value of current is I through three channels. The current which is being generated is I. So is equal to 3I. Okay, so this can be summed together. This is the current which is being generated through this membrane. Okay, so they saw that the whatever single ion channel generates, the second ion channel and third ion channels, all of those are summed together to give the current which is flowing through the membrane. Okay, so this means that the value of, the value of G, the conductance of postsynaptic membrane, which has, let's say multiple acetylcholine receptor, let's say it has 6 acetylcholine receptors. So this will be equal to 6 multiplied by conductance, which is due to the single ion channel, single acetylcholine receptor. So the G is equal to N into gamma. Gamma is the conductance due to single ion channel. while N is the number of ion channels, means number of acetylcholine receptors that are present on the membrane. So in this case, here are the three, here are let's say here are three ion channels. So if the conductance value, the total conductance will be 3 into the conductance due to single ion channel. Okay, so this was proved by this experiment. Okay, by this recording proved that the each ion channel current can be summed together to give the total ion current. Okay, so each ion channel, so the each acetylcholine receptor is going to produce 2 - 2.78 nanoampere, sorry, not nanoampere, picoampere, minus 2.7 picoampere current, and multiple ion channels, usually they generate the membrane potential, the current, the value of current is minus 900 nanoampere. Okay, so this much. So there are almost 2 lakh 2 lakh acetylcholine receptor is present on a muscle fiber, on a patch of muscle fiber, and they produce minus 500 nanoampere current. Okay, single acetylcholine receptor produces how much? Minus 2.7 picoampere, and whole multiple they produce minus 500 nanoampere current. Okay, so this was seen by those scientists. Well, this is another recording which tells you about the smooth decay of the current. Okay, so what happens is that whenever acetylcholine pulse is given, it can, generate, it can open the ion channel for multiple, like for different durations. Here you can see it is open for different duration. Okay, but whatever current is being generated, the current value of current is same. Here you can see. Okay, this is how the rectangular ion currents are generated by the channels. So this diagram shows that on a receptor, on a membrane, there are 6 ion channels. Okay, so 6 ion channels are present, and this is how this is what they are producing after being activated. Okay, look, like let's say that this. Oh well. Oh, where is it? Mhm. So this experiment was something like this. On There is a membrane. Mhm. Okay, so there is a membrane. Hmm, On that, 6 acetylcholine receptors are present. Okay, 1, 2, 3, 4, 5, and 6. Okay, so when acetylcholine is introduced in this system, let's say that acetylcholine bound to them, and whatever current is being generated in by individual acetylcholine receptor, that was measured, and that was seen like this. So let's say that the current which is being generated by the channel 1, it is, it is I. By 2, it is also I. By 3, it is also I. 4 also generates I. 5 also generates I. And 6 also generates I. But the duration for which I is generated, let's say that this is 1 millisecond, this is 2 millisecond, let's say this is 1.5 millisecond. So whatever duration was that, it was different. Okay. And so due to so even though the ion channels, they are producing same current, but why does that current dissipates? That current has a smooth decay, because all of those currents, they are being generated for different time. Okay, so for different time frames. So this shows 2 millisecond, and the scale is 2 milliseconds. So here, let's say that for when you take a look at the total ion current that is flowing through this membrane, you see a smooth curve, a smooth decay curve, just like a radioactive decay. Okay, you see that the current dies out smoothly, and this is due to the generation of same amount of current for different time duration. Okay, I hope I'm clear to you all. So this smooth curve, this dying out current that is seen, this dying out current, here you can see, this is seen due to the opening of ion channels producing same current, but that opening is for the different duration for some ion channels are open for long, some ion channels are open for less time. Okay, so due to this, a smooth decaying curve is seen, for the total ion current. So this almost follows all the statistical law that are followed in the radioactive decay. Okay, we'll learn about this, the analogy between that and the opening of ion channels. So this opening of ion channels is random, but it is not, it is actually stochastic. Means they follow the same statistical law. Well, they follow some statistical law. So it is random, but it is coordinated. So it is stochastic. So they follow the uh, same statistical law that is seen during the radioactive decay. So we'll discuss about this in next lecture. And in next lecture, we are also going to discuss about the properties, the molecular properties that are exhibited by the acetylcholine receptor. So in this lecture, we only discuss about all of the experimental details, the experiments that were done on the neuromuscular junction that made us to reach on the stage. Like, how do you think that we, whatever we said, that cell this membrane is polarized, it undergoes depolarization, then undergoes repolarization, and then after hyperpolarization, it achieves its polarized state. It comes back to the resting membrane potential. This cycle of action potential generation and propagation, this was given by the experiments that were made like this. So like, how did we deduce that only ion, which ion current is being generated during which stage of the cycle? That was deduced by those experiments. So just to know that, just that you should know about the experiments that were done by researchers, I made this lecture. And this was the second part of the previous, like, neuromuscular junction series. In next lecture, we will learn about the molecular techniques, molecular properties of acetylcholine receptor. And with that, we will be done with the whatever studies were done on the neuromuscular junctions, which due to which the field of neuroscience developed. Till then, see you. Bye bye.